Welcome to the lecture 6 of recombinant DNA technology. Today we will be discussing about transgenic animals and the process of generating transgenic animals, and then brief discussion about the cell culture. So transgenic animal is the one that has integrated a gene or a DNA sequence into the genome of the cell, and it has been done by the intervention of human subject. So that means human, we are actually doing or transferring a foreign DNA sequence or a gene into another animal. So after transferring the gene can be incorporated into the germline, and it's able to actually pass to the future generations. It has been extensively used, this technique, in the biotechnology for studying new or modified genes and identify the functions of that gene. So it is actually a bit precise technique than the crossbreeding, but eventually the result is similar to the crossbreeding. So there are many potential applications of this generation of transgenic animals. For example, this method can be used to develop a new or improved livestock or agricultural products like crops. We know that's the GMO, genetically modified products. And for the livestock, it can be used to improve the reproductive performance or the increased can be feed utilization or the growth rate, increase the production or disease resistance, this kind of matter. In case of medical importance, the transgenic animal has been extensively used to obtain the information of a function of a genes or the regulation of human diseases. It is also used to get a high value products in recombinant pharmaceutical proteins, like the recombinant pharmaceutical proteins, which can be used in human therapy, like the insulins. So gene can be transferred to another host using various techniques, such as microinjection techniques or the embryonic stem cell mediated gene transfer or the retrovirus mediated gene transfer. We will briefly go through each of these three techniques, that how actually they can be processed to generate a transgenic animal. So since its inception in the early 90s, the micro injections or the needle micro injections is a prominent experimental approach in biological research to transfer the gene to another organism. And it is actually used to transfer cellular organelles, DNAs, RNAs, even enzymes or proteins. So in this process, the foreign gene is actually injected into the male pronucleus of the fertilized egg. So the pronucleus is actually the nucleus of a sperm or an egg cell during the process of a fertilization. So the sperm cell becomes a pronucleus after the sperm cell enter into the ovum, but before the genetic material has been fused with the egg. Contrary to the sperm cell, the egg cell has a pronucleus. Once it becomes haploid, we know that the somatic cells, they have contained 46 chromosomes, and they are the diploid cells. And the germline cells, like the sperm and egg, when they, during the reproductive process, they actually do the meiosis and produce a haploid that is half of the 46 chromosomes that they normally contain 23 chromosomes. And when the male and the female pronuclei or the genetic material has been fused together, so they actually then result a single diploid nucleus, which is the embryo, and that embryo actually contains the full set of 46 chromosomes. So in case of the micro injections, the transgene actually injection has been performed by holding the fertilized egg with a small amount of suction, and then using another micropipette to inject the desired gene into the pronucleus. And in the next stage, that pronucleus has been implanted into the pseudo-pregnant surrogate mother. The pseudo-pregnancy is just creating a false pregnancy. So it has a physical state where it contains all the signs and symptoms of pregnancy without the presence of a real fetus. So that in that way, the gene has been transferred to like the mice or mouse and grow them together. So in case of embryonic stem cell mediated gene transfer, we have just seen in our previous slides that when the haploid sperm and the egg cell fuse together or fuse the genetic material together, they contain a diploid nucleus which contain that 46 chromosomes and produce an embryo. And this embryo can be formed by in vitro fertilization, and this embryo then grown for a few days in a stage that's called a blastocyte. And in that blastocyte, there is a cell called inner mass cell, and the embryonic stem cells are then isolated from that inner mass cells of the blastocyte. So this is the inner mass cell. The embryonic stem cells has been isolated from there, and these embryonic stem cells then manipulated to take up the foreign DNA, either using a micro injections technique or using a viral vectors or any other physical or chemical transfection like the liposuction using a lipid molecule. So embryonic stem cell is a very, very powerful tool to analyze the gene functions and the regulations, because these cells can be manipulated to form any kind of cells. So the inner mass cells or the embryonic stem cells, which is actually have potential to grow in any kind of cells, such as it can be a brain cell, can be a cardiac cell, can be a muscle cell, etc. and etc. So it can give a specific signaling molecule and a growth condition, and then use these embryonic stem cells to grow as a brain cell or grow as a cardiac cell. That's why this cell has been called pluripotent. So after manipulating the embryonic stem cells, they are then injected back into the blastocytes, and the embryo is then implanted into the uterus of a recipient, which can be mice. So this is the process of developing a embryonic stem cell mediated transgenic. So the other technique is the retrovirus mediated gene transfer. And we know that in nature that viruses have the ability to infect a host cell and push their genetic material into the host cell that can be integrated into the genome of that host cell. And the great example is we know about the COVID-19. So the SARS virus is actually inserting that genetic materials to the human, and then from there that gene has been integrated into the system of all the cells, and then they are growing. So these characteristics of the virus can be efficiently used to deliver a specific gene into a host organism. And the most commonly used viruses for the gene delivery are the retroviruses, because they integrate the viral DNA directly into the chromosome. There are viruses such as adenoviruses and lentiviruses. They can be used to transfer the genetic material into the host organism. And I'm sure that we will be discussing this in another chapter, which is called the gene therapy, in a detailed fashion. So similar to the molecular cloning, when we transfer the gene to another organism or generating a transgenic animal, we definitely need to screen that animal, whether that animal has actually taken up the gene, number one, and number two, whether the gene is expressed the proteins. So it is very important to know that the protein is also expressing, because the function is actually coming from the protein. So usually Southern blots or PCR can be used to determine the presence of the gene. So PCR is a very simple technique, as we discussed in our last lecture. So we can get the genomic DNA or extract the genomic DNA from the animal. We can be use a little bit of small tissues or the plants, and then extract the genomic DNA from the transgenic animal. And using a gene-specific primer, we can use that genomic DNA as a template and do a PCR, and then identify whether our gene is actually present or not. Similarly to PCR, we can also use the Southern blot. If we can recall that we extract the genomic DNA and then digest them with a specific restriction enzyme, which we know that this restriction enzyme can cut into the specific gene size of the desired gene, and then this this digested product has been transferred to a membrane, hybridized into the membrane with a probe, specific probe and then detect by autoradiography to check whether we have a specific band of that gene. And southern blot again will tell us how many copies of that gene actually integrated into the chromosome. For detecting the protein expression, after we confirming that our gene is present, the next step is whether that gene can produce that protein. So in this case, Western blot, which actually detect the protein, so we can extract the protein from the cells, and that protein can be detected against a specific tag or epitope tag to identify that the protein is expressed. We can also do ELISA, which is an enzyme-linked immunosorbent assay, which also tell us that the protein has been expressed.